मैम स्टार्ट कर दूं?
Good morning, everyone. First of all, thank you all for finding time to join the virtual international conference on Biotrendz Advancement and Challenges in Science and Technology, organized by Department of Biotechnology, Noida Institute of Engineering and Technology, Greater Noida. My name is Hina Pahwa. I am Assistant Professor at Department of Biotechnology, NIET, Greater Noida. NIET is a premier engineering and management institute and in NCR region of India. The mission of the Department of Biotechnology at NIET is to evoke a zeal in young minds to gain technical, analytical, and employability skills in the field of life sciences and biotechnology. As a part of this mission, we have organized this two-day academic conference in order to broaden our knowledge and perspective of the field. The conference is organized in association with Swadeshi Science Movement of India, which is a non-profit organization working for science popularization and implementation of modern technology and ancient sciences in India. Today, we will have three invited talks by esteemed guests from 11:05 to 12:15 p.m. Then, after lunch break, we will continue with the interesting research talks by research scholars from 2 to 4 p.m. Tomorrow also we'll have invited talks by eminent scholars in morning session and poster presentation by students after lunch, followed by declaration of results and certificate distribution. In Indian culture, we revere goddess Saraswati as giver of knowledge, wisdom and consciousness. So let's start the conference by offering a prayer to Ma Saraswati. That was so soothing. Now, without further ado, may I request Dr. V.M. Kapse, Director NIET, to give a welcome address to all the invitees and participants. Sir, please.
So you need to unmute, please.
Good morning. Namaskar, madam. Honorable Chief Guest of today's function, Srimati Nilima Katiyar, madam, Minister of State, Higher Education in the Government of UP, Shri Raman Batraji, Executive Vice President, NIT Greater Noida, Dr. D.P. Bhatt, President, Swadeshi Science Movement, Delhi, Shri Praveen Sonejaji, Director General, NIT Greater Noida, Dr. Abhijit Mazumdar, Director, Pharmacy Institute, Dr. Lakshmi Mishra, HOD Biotech, all the guest speakers from India and abroad, other head of the departments, faculty members and participants. On behalf of NIT, I am honored to welcome the chief guest of today's function, Honorable Srimati Nilima Katiyar, madam. We are thankful to you, madam, for taking out your valuable time and gracing us on this occasion. Your word of wisdom will be a great motivation for all of us. I am delighted to welcome our visionary, dynamic Executive Vice President, Shri Raman Batra, sir. It's my pleasure to welcome Dr. D.P. Bhatt, sir, and we are thankful to you, sir. Bolo, bolo. Sorry for inconvenience due to some technical problem. I was disconnected. Uh, I would like to welcome all the speakers, guest speakers, participants, and everybody. NIT, talking about the NIT is one of the premier institute of engineering, pharmacy, and management of India's National Capital Region. BTech program in computer science engineering, mechanical engineering, biotechnology, information technology, and B pharmacy are accredited by NBA. Also, institute is NAC accredited with a CGPA of 3.23 with A grade. NIT has been ranked in the band of 201 to 250, and Institute of Pharmacy ranked under 50 by NIRF 2020, one of the prestigious rank by MHRD. Also, Institute has received E-Lead certificate by QS iGauge ranking for its outstanding contribution in e-learning. Institute is ranked in top 50 institute by Atal ranking of institution on innovation achievements. I'm happy to share with you that NIT has got the autonomous status, and it is the first private institute in UP who got this autonomous status. On this occasion, I would like to congratulate the Dr. Lakshmi Mishra, HOD Biotech, and the entire biotechnology department for organizing the international conference on Biotrendz, advancement, challenges in science and technology. The coronavirus disease 2019 pandemic has presented some significant challenges to the scientific community. However, this has also offered opportunities for pursuit of new scientific activities, and in particular for the field of biotechnology. Definitely, this conference will provide an opportunity to all the participants as well as speakers to exchange their thoughts and to evolve a new technology which will be definitely beneficial to find out the solution of this coronavirus. On this occasion, once again, I would like to congratulate the Department of Biotechnology. Also, I would like to welcome all the dignitaries. Thank you.
Thank you so much, sir. Now, may I invite Dr. D.P. Bhatt, the President of Swadeshi Science Movement, Delhi, to address the participants and inaugurate the conference. Dr. D.P. Bhatt, sir, please.
Namaskar. Are you hearing me?
Yes, sir.
Namaskar. Good morning.
Good morning, sir.
माननीय राज्य मंत्री श्रीमती नीलिमा कटियार जी, श्री प्रवीण सुनेजा जी, डायरेक्टर जनरल NIET, डॉक्टर VM कापसे, डायरेक्टर, डायनेमिक डॉक्टर रश्मि मिश्रा, श्री रमन बात्रा, invited speakers from within the country and overseas. I am really humbled to be here amidst you all, amidst this get together to welcome you all in the international conference on Biotrendz, advancement in science and technology, organized by NIET in association with Swadeshi Science Movement of India, also called as Vigyan Bharati Delhi. If you look at my slide, just one slide I have just made it, which is very important. I will start from the quote of the Honorable Prime Minister, who gave a statement in the year 2017. India's S&T will be one among the top 3 nations in the world by 2030. This followed another statement recently in the lockdown 4 address, where he talked about the self-reliant Bharat and prompted us to be vocal for local towards new development model. Friends, it's not possible. Just Prime Minister's saying will not serve the purpose until and unless we bring in all the components of indigenous sciences in our policies, be it in science policy, education policy, technology policy, or any other policy that talks about the development of this nation. And for this, I just decided to look at some 5 key points. If time permits, I will speak on these 5 points, maybe 2-3 minutes each, or if you think that time is over, you can stop me anytime. And these 5 key points, I will just talk about brief on SSMD and what should be our sort of वैज्ञानिक जीवन दृष्टि जिसको हम कहते हैं, जो इस समय हमें देखते हैं कि विज्ञान को हमने टेक्नोलॉजी से भी उसका कंपैरिजन कर रहे हैं। विज्ञान को आप हम लोग बहुत नीचे पाएंगे, आज इस तरह नीचे जा चुका है। तीसरा, this is very important how we communicate with each other in science and particularly in this country, whether we depend just solely on English or there a need to have a communication through our own languages. And 4th point, sometimes we just come and give the lecture and get away from that. I personally believe that unless otherwise you contribute, you have contributed, you have no right to speak. So the 4th point is this COVID time, this unfortunate time for the country, and it has come as a boon for us because we have contributed a bit on that, maybe 2-3 lines. Then finally, intellectual property. It's a very important subject. It has to be very broad. And the new national IPR policy, which has come in the recent years, how it is relevant in the present perspective, and how important is the Rishi tradition, our ancient wisdom and all that. And also we can look at it from the point of view of 17-point UN agenda, and ultimately from the common point of view, common man's point of view. The first one, brief on SSMD. The Swadeshi Science Movement is an all-India organization working independently in each state, including Delhi. It's one of the most positive science movements in the country. For the first time, the spirit of India, ethos of India, and culture of India is taken as the base for development. And we took the beautiful statement from Isavasya Upanishad as our motto, well depicted in the logo as you see in the slide itself. Avidya Mrityum Tirtva Vidya Amritam Asnuti. Yani, with material and physical sciences, we fight for the causes of unnatural death, while with spiritual sciences, we attain higher stages of consciousness, that is immortality. Accordingly, friends, we took the combination of all 3, that is spiritual sciences, material sciences, and social sciences, for the concurrent and harmonious development, which has been reflected in our workings over the years to have promoted these aspects in the innovations and also the village development programs over the years. And next one is, as I said, वैज्ञानिक जीवन दृष्टि, कि हमने विज्ञान के प्रति ध्यान हम लोगों का काफी नीचे जा चुका है. Is there a need to just overcome that now? अंग्रेजी में कहें तो scientific vision of the life that our rishi or ancestors used to have. I will just give you a very important example. जो सदियों से चल रही हमारी भारतीय अवधारणा, जो आज बिल्कुल उल्टी हो गई है, rather it was very relevant in the earlier years, और हम लोग विनाश की ओर अग्रसर हो रहे हैं, और कोविड ने भी अपना इसमें गोता खाना शुरू किया है. और ये अवधारणा हमारी ये रही है कृषि के संबंध में, उत्तम खेती, मध्यम बान, अधम चाकरी, भीख निदान. कृषि को हमने सर्वोत्तम कार्य माना था, क्योंकि उसमें किसान एक बीज से 1000 बीज तक का उत्पादन करता था, तो उसमें साइंस इज हिडन वेरी मच इन दैट. मध्यम बान, व्यापार, आज किसान ये सब भूलकर छोटी मोटी नौकरियों के पीछे भाग रहा है. I am just trying to relate with what Prime Minister said about Atmanirbhar Bharat. It cannot just happen unless we understand, realize what was their great in our ancient wisdom. और जो भी हमारी भारतीय अवधारणा थी, क्या वो सही थी या गलत थी? तो अब हम किसान नौकरियों की तरफ भाग रहा है. अधम चाकरी, नौकरी करना प्रतिष्ठा का विषय बन गया है. हर व्यक्ति बीटेक करता है, एमटेक करता है, उसके बाद एमबीए करके दुकान चलाता है या बिजनेस करता है. देयर इज नो पॉइंट इन देन डूइंग बीटेक, और ही बिकम्स आईएएस एंड फॉरगेट्स द साइंस. तो हमने नौकरी करना प्रतिष्ठा का विषय बना लिया है, जिसको हमने निष्कृष्ट मतलब सबसे नीच कार्य कहा गया था हमारी अवधारणा के हिसाब से. और हमने जुगाड़ टाइप चीजों पर भी ज्यादा ध्यान नहीं दिया कि भारतीय लोग जुगाड़ में बहुत मशहूर होते हैं. वी यूज्ड टू जस्ट मेक अ फन ऑफ इंडियंस. आई थिंक दैट्स नॉट करेक्ट. जुगाड़ में भी साइंस इज हिडन. तो जुगाड़, इनोवेशंस, and particularly with regard to the informal sector. Informal sector, I mean grassroots innovators, artisans, tribals, and farmers. We will have to give the relevance to them, and it's the responsibility of the formal sector, where you have a institution, a great institution like NIET also has got a greater responsibility. And I will give another example. When we relate विज्ञान with technology, मोबाइल टेक्नोलॉजी का यदि आप उदाहरण लें, हर महीने टेक्नोलॉजी बदलती है. हर तीसरे महीने, दूसरे महीने आपके पास नया मोबाइल आता है. डू यू थिंक देयर इज अ वेरी ग्रेट साइंस इन दैट? इट्स नॉट. एक्चुअली 19वीं शताब्दी के बाद वायरलेस टेक्नोलॉजी का विस्तार हुआ, साइंस का विस्तार नहीं हुआ. और मार्कोनी साहब, उन्होंने ये सबसे पहले ये चीजें इजाद की थी. लेकिन वी फॉरगेट दैट डॉक्टर जगदीश चंद्र बोस, he has done the science part of it during 1894-96, and even he had taken the patent on that. और डॉक्टर जेसी बोस साहब ने पेरिस सम्मेलन में एक शताब्दी पूर्व एक्सपेरिमेंटली डेमोन्स्ट्रेट करके बताया कि जिस प्रकार मनुष्य के अंदर प्राण तत्व है, उसी प्रकार वनस्पति में भी जीवन होता है. और यह यंत्र निर्माण करके उन्होंने सिद्ध किया. डू यू नो, यू आर ऑल बायोटेक्नोलॉजी पीपल. ही इज अ पर्सन ऑफ फिजिक्स, डॉक्टर बोस, बट ही हैज डन द वर्क ऑन बॉटनी. हाउ इज इट पॉसिबल? It's just possible. He had all the great knowledge of our Upanishads, which he had attained by meeting Swami Vivekananda in the year 1900. He had a thorough knowledge about Upanishad. That's how, being a physics man, he was able to contribute greatly in the area of botany or biotechnology. Next, friends, he did one experiment there in the Paris Sammelan. Perhaps some of you may be aware. He wanted to prove that metals में भी जान होती है. और उन्होंने क्या किया, सबके सामने साइनाइड को आयरन रॉड में डाला. और he expected that it will lead to a sort of vibration in the iron rod. कांपने लगेगा आयरन रॉड. लेकिन जब उन्होंने एक्सपेरिमेंट किया, ऐसा कुछ भी नहीं हुआ. आयरन रॉड रिमेन्ड पैसिव. सो इवन डॉक्टर बोस सकते में आगे थे. सारे लोग हंसी मजाक बनाने लग गए थे डॉक्टर बोस की. बट ही वाज सो कॉन्फिडेंट. उन्होंने साइनाइड पी लिया. इन द नेक्स्ट मोमेंट दूसरे लोग सकते में आ गए कि डॉक्टर बोस को कुछ भी नहीं हुआ, क्योंकि वो किसी की कंस्पिरेसी थी. साइनाइड की जगह उन्होंने पानी मिला दिया था. फाइनली साइनाइड वाज ब्रॉट बैक एंड दैट आयरन रॉड स्टार्टेड वाइब्रेटिंग. पॉइंट आई एम मेकिंग हियर इज दैट जो साइंस तब जगदीश चंद्र बोस साहब ने दिखाई थी, वो आज भी इट इज स्टैंड स्टिल टिल डेट. देयर इज नो फर्दर प्रोग्रेस ऑन दैट. That's what I wanted to just highlight. There is a need to bring about a sort of scientific temperament among the youths now, hereafter. Third, Bani ka Vigyan. It's very important. Friends, there is a myth that Indian languages are no good for science communication, for which English alone has to be used forever. It needs to be revised in order to take all necessary steps to popularize science amongst all sections of society. If we really want science to be mass commodity and not class commodity, all regional languages are to be used as effective media for science communication. That is the first component of Swadeshi education, is to inculcate a sense of love for its own languages, rather mother tongue and the common language Hindi. And I am proud to say that SSMD has been able to contribute in that through bilingual mode, even multilingual mode, Hindi and English, without opposing English, we have been able to contribute. We brought out more than a dozen publications in that. It's very unfortunate. यदि किसी यूथ से पूछा जाए 69 क्या होता है, तो वो पूछेगा is it 69? और 89 यदि कहेंगे वो पूछेगा इज इट 79? आई मीन हम सोचते हिंदी में है लेकिन करते अंग्रेजी में है. That's how we are unable to do great innovations or great developments, and creativity is missing. Creativity really gets missed क्योंकि आप सोचते अपनी भाषा में हो, करते अंग्रेजी भाषा में हो. And I'm again proud to say that in CSR, where I was the part for 37 years, 1998 they brought out a policy that we should do work on Hindi as far as science and technology is concerned. But in SSMD, we brought out a book in 1997 itself, before the policy came. It's so sad in this country, policies are made by Honorable Prime Minister or the Cabinet or the Parliament, but who implements that? So it's our duty, foremost duty, to do things the way it suits us. At least after the Corona time, there is an opportunity to learn. And next point, what is that we did in the COVID time? I'm just lecturing here, or have you contributed? Yes, पहली बार हमने कोविड महामारी काल में सिर्फ प्रतीक से डिजिटल स्वदेशी विज्ञान पत्रिका का प्रकाशन जुलाई 2020 में किया, और उसका फोकस था कोरोना एंड एनवायरनमेंट. और इसी कोविड काल में आदरणीय प्रधानमंत्री द्वारा उनके लॉकडाउन 4 संभाषण में लोकल व स्वदेशी मंत्र पर बल दिया गया. वह आत्मनिर्भर भारत बनने का नया विकास मॉडल हमारी मोटर ऑपरेंडी को राष्ट्रीय अंतर्राष्ट्रीय दोनों स्तर से अधिक प्रासंगिक बनाता है. यदि आप लोगों को रुचि हो तो यू कैन काइंडली मेल मी एंड आई विल सेंड यू दैट पब्लिकेशन व्हिच इज अवेलेबल विद SSMD. एंड अनदर एग्जांपल आई विल गिव यू, क्योंकि आप लोग बायोटेक्नोलॉजी के विद्वान हैं, छात्र हैं, तो आपको पता होगा कि भारतवर्ष में सितंबर माह में पोषण माह के रूप में सितंबर माह को मनाया जाता है. हाउ मेनी ऑफ यू सेलिब्रेट दैट एंड हाउ मेनी ऑफ you know about that? I don't know. But SSMD, in collaboration with Vinoba Bhave University Hazaribagh, has done a wonderful work last month in September, and they took the topic strengthening nutritional garden theme this time, and organized a 3rd national conference just on 27 September. और उन्होंने awareness camps organize करे in the Hazaribagh Jheel area, and also in the Jhumri Taliya Ashram Road, same in Hazaribagh, and distributed handouts and lagwahar sort of booklets regarding how to go about in dealing with स्थानीय फल, स्थानीय सब्जियां, इनका कैसे आप भरपूर उपयोग करेंगे. और उसमें उन्होंने मास काउंसलिंग करी जिससे कि लोगों की रोग प्रतिरोधक क्षमता में इजाफा हो. That's what we have contributed in the role. And finally, I'm sorry, I think if I'm taking more time, I can stop. You can stop me anytime. And how important the IP, intellectual property, and new IPR policy and receipt tradition, and also the 17-point UN agenda is important. And it's important with reference to the common man. Or all the research programs has to be common man centric. That's what the UN 17-point agenda says, and that's what the IPR policy of the government says. Gone are the days, friends, when we have had projects like convenient luxury, and we used to just publish them to get our promotions and other upliftments, and to raise our biodata. In other way, we used to think in the individual mode. But now, as per the policy of the government, time has changed. You have to think about the society. It's better that you look at the problem of our R&D after looking around the problem in the periphery and see the pain of the society and work in that painful area. Because new IPR policy is commendable for taking note of our informal sector, informal economy, as I said earlier, about artisans, farmers, tribals, grassroots innovators, who has to work on that. So there is a need to revisit every nook and corner of the villages in the country. With these few words, I would once again welcome you all. Thank you very much. Namaskar.
Thank you so much for your words of wisdom and enlightening us with interesting facts. I would now like to invite Dr. Lakshmi Mishra, Head of the Department of Biotechnology and the convener of the event, to introduce the conference to all of us. Ma'am, please.
Thank you. Thank you so much, Henna. A very good morning to all and very good evening at the respective places. On the behalf of Department of Biotechnology, I extend a warm welcome to our chief guest, Honorable State Minister, Srimati Nilima Katiyar ma'am. Ma'am, welcome. And thank you so much for taking out time from your busy schedule to grace the occasion. I'm grateful to our Executive Vice President, Raman Batra sir, for his support and presence. President Swadeshi Science Movement, DP Bhatt sir, for his motivational talk. Our Director General Praveen Suneja sir, Director sir, Vinod Sapsi sir, for their continuous guidance. I welcome all the invited speakers and delegate to the conference. Today, I take a pride to share that moving forward with time, this Biotrendz conference is the second of the series, and we have taken it to an international level. The conference will provide a platform to achieve the prevailing gap in transformation of multidisciplinary science. The theme of the conference, Advancement and Challenges in Science and Technology, covers a large area from medical healthcare, indigenous science, environmental biotechnology, nanobiotechnology, new age drug therapy, green technology, computational biology, Internet of Things, and artificial intelligence. Because we can see these days biotech everywhere from the era of Vedic period, as just discussed, to the present COVID pandemic. I'm overwhelmed to share that we got a wonderful response, not only from India, but across the globe, and we got around 300 plus participation requests. After scrutinizing, we could reach 200, but due to the limitation of time, we could select a handful from the pool to be expressed in terms of presentation in the conference. This conference will continue for 2 days, 9th and 10th, and will have 5 sessions. In first and third session, we are having invited speakers across the globe, and in third, fourth, and fifth session, second, fourth, and fifth session, established and budding researchers will express their work. The fifth session will be followed by a valedictory, where we'll be announcing the best presenters. I assure to all the participants and the audience that 2 days will be full of knowledge sharing, and everybody will get good food for grey matter. The team Biotech has worked very hard to make today happen, and my bestest wishes to my team. With this, I declare the conference open. Thank you so much.
Thank you, ma'am. Indeed, it is a very proud moment for all of us. So now I request Mr. Praveen Suneja, Director General, NIET, to address us all with his valuable words. Sir, please.
Good morning.
Good morning, sir.
सबसे पहले श्रीमती नीलिमा कटियार जी का अभिवादन और धन्यवाद कि आप एक बार हमारे साथ दोबारा जुड़े और हमारा मार्गदर्शन करने के लिए आपने समय निकाला। डॉक्टर डीपी भट्ट, मैंने आपको सुना और पहली बार डॉक्टर रश्मि मिश्रा द्वारा दिए गए ब्रोशर के थ्रू मुझे आपकी स्वदेशी साइंस मूवमेंट के बारे में पता लगा और मुझे ये भी पता लगा कि आप किस तरह का अच्छा काम कर रहे हैं। आपका भी धन्यवाद। I would like to thank all the participants and eminent speakers from all over the world who are going to grace and talk about this subject over next 2 days. I'm also thankful to Dr. Lakshmi Mishra, entire leadership team of NIT and the organizing committee for stitching this conference together. Such exchange of views across the world, beyond the boundaries of a country, definitely help not only in sharing the knowledge, but also to build on each other's idea. And once we do that, it leads to advanced knowledge sharing and a spiral which is upwards. And I am sure all the participants are going to be greatly benefited over next 2 days. With these words, I thank you all and wish the organizers and the participants all the best. Thank you very much.
Thank you so much, sir. So may I now request Mr. Raman Batra, Executive Vice President, NIET, to further uplift the spirits of all the participants by his inspiring words. Sir, please.
Raman, you are on mute. Raman, you are on mute. Unmute karo.
A very good morning to all. हमारी आदरणीय राज्य मंत्री श्रीमती नीलिमा कटियार मैडम को सादर प्रणाम और मुझे बड़ी हार्दिक खुशी है कि मैडम ने दोबारा हमें ज्वाइन करा और बड़ा अच्छा लगता है कि अगर जो स्टेट गवर्नेंस है वो इतनी प्रोएक्टिव है तो कॉलेजेस को भी उतना ज़्यादा प्रोएक्टिव होना। डॉक्टर भट्ट ने बहुत ही अच्छा बोला कि जो साइंस और टेक्नोलॉजी है हमारे प्रधानमंत्री जी का विजन कि हम 3 देशों में होंगे 2030 तक, तो मुझे पूरी आशा है कि हम रहेंगे। मैं आप सब लोगों का ध्यान कुछ टेक्नोलॉजीज़ पे लाना चाहता हूँ, बायोटेक्नोलॉजी से रिलेटेड, क्योंकि इस टाइम कोविड के समय में ये एक ऐसी ब्रांच है, ऐसा विषय है जिसकी इम्पोर्टेंस बहुत ज़्यादा है। तो मैंने 6 चीज़ें सोची जिसके बारे में मुझे बात करनी चाहिए और बहुत इम्पॉर्टेंट है। पहली है बायोसेंसर्स। मैडम, हर जगह आपने देखा होगा कि लोगों का टेम्परेचर रिकॉर्ड हो रहा है, ऑक्सीजन लेवल रिकॉर्ड हो रहा है। तो ये बायोटेक्नोलॉजिस्ट हैं जो हर डिवाइस में बायोसेंसर्स लगा सकते हैं, जिसके द्वारा हम पल्स, जिसके द्वारा हम ऑक्सीजन, जिसके द्वारा हम टेम्परेचर नाप सकते हैं। तो बायोसेंसर एक ऐसी चीज़ है जो आने वाले दिनों में हर इलेक्ट्रॉनिक डिवाइस में, जैसे भट्ट साहब ने कहा, वो होगा और बायोटेक्नोलॉजिस्ट उसमें बहुत बड़ा रोल प्ले करेंगे। दूसरा मैम विषय, हिंदुस्तान जाना जाता है मेडिकल टूरिज्म के लिए। विदेशों से लोग आते हैं यहाँ पर इलाज कराने, लेकिन एक बहुत बड़ा एरिया जो निकल के आ रहा है वो ट्रांसप्लांट का है, जिसमें हार्ट ट्रांसप्लांट, किडनी ट्रांसप्लांट, लिवर ट्रांसप्लांट। उसके अंतर्गत 3D प्रिंटिंग जो आज की डेट में सिर्फ पार्ट्स की होती है, आने वाले दिनों में जो ऑलरेडी कुछ कंट्रीज कर भी रही हैं, उसमें मेरे ख्याल से हमारे देश को लीड लेनी चाहिए। और हमको 3D प्रिंटिंग जो बेसिकली बायो प्रिंटिंग कहलाती है, जिसमें एक इंसान के टिश्यूज से उसी का पार्ट डेवलप किया जा सकता है, वो एक और टेक्नोलॉजी है जहां बायोटेक्नोलॉजी का बहुत बड़ा कंट्रीब्यूशन है। तीसरा, हम सबको मालूम है सर्दियां आ रही हैं, दिल्ली के आसपास स्मॉग हो जाता है, पॉल्यूशन। गवर्नमेंट का भी बहुत फोकस है कि प्लास्टिक्स को बैन किया जाए। यूपी गवर्नमेंट ने बहुत अच्छे स्टेप्स लिए। हमारे मुख्यमंत्री जी ने बोला कि हमेशा एनवायरनमेंट, पर्यावरण का ध्यान रखा जाए, बहुत पेड़ लगाए जाए। बट बायो प्लास्टिक्स जो है वो आगे का फ्यूचर है। डिग्रेडेबल मटेरियल्स का यूज इस देश को विश्व की तरह ले जा सकता है, जहां पर बायोटेक्नोलॉजिस्ट दोबारा बहुत बड़ा रोल प्ले कर सकते हैं। चौथा एरिया बायो एनर्जी है। आपको मालूम है कि धीरे धीरे हम इलेक्ट्रिक व्हीकल्स पे शिफ्ट हो गए हैं और हो रहे हैं। इसी तरीके से बायोफ्यूल्स जो बड़ी बड़ी इंडस्ट्रीज में यूज हो सकता है, उसको बहुत इंपॉर्टेंस है। पांचवा एरिया जीन एडिटिंग। मैडम, जैसे हम मेडिकल टूरिज्म में नंबर 1 है, आने वाले दिनों में हमारे बायोटेक्नोलॉजिस्ट उस लाइन में काम कर रहे हैं। और जैसे हम ऑटोनोमस हो गए हैं, अपने ऑटोनोमस सिलेबस में भी हमने जीन एडिटिंग के बारे में पढ़ाया है। आज से 10 साल पहले जीन एडिटिंग जो होती थी वो करीब करीब 95 मिलियन डॉलर्स की कीमत होती थी, जो आज की डेट में करीब $1000 के आसपास कीमत रखती है। तो जीन एडिटिंग वो जहां पर हम डिफरेंट जीन्स में ला के एक बेहतर प्रोडक्ट ला सकते हैं। और चौथा और आखिरी AR VR। जैसे हम सब लोग डिजिटली कनेक्टेड हैं, ऑगमेंटेड रियलिटी और वर्चुअल रियलिटी बहुत इंपॉर्टेंट है। जहां पर मेडिकल कॉलेजेस में, जहां पर कॉलेजेस में एक सिर्फ डेड बॉडी मिलती है काम करने के लिए डॉक्टर्स को, AR VR वो रेवोल्यूशन है जहां पर बच्चे बार बार हार्ट की सर्जरी कर सकते हैं, किडनी पे वर्क कर सकते हैं, डिफरेंट पार्ट्स पे वर्क कर सकते हैं। तो ये 6 एरिया ऐसे हैं कि जहां पर एडवांसमेंट बहुत हो रही है। और जो आज का टॉपिक है बायो ट्रेंड्स इस कॉन्फ्रेंस का, वो बहुत ही अच्छा टॉपिक है। मेरी हार्दिक शुभकामनाएं हेड ऑफ डिपार्टमेंट बायोटेक्नोलॉजी और सारी टीम को कि बहुत अच्छा टॉपिक यूज़ किया है। और जो हमारे माननीय मुख्यमंत्री जी इस उत्तर प्रदेश को आगे बढ़ाने के लिए भरसक प्रयास कर रहे हैं। मैडम, मेरी आपसे गुजारिश है कि अगर हम एक इस प्रदेश में बायोटेक्नोलॉजी पार्क की कल्पना करें, जो आज की डेट में पुणे और बेंगलोर में है, मेरे ख्याल से आने वाले दिनों में बहुत बहुत बड़ा सेक्टर हो सकता है। हम दुनिया के सबसे बड़े टैबलेट मैन्युफैक्चरर्स हैं, फार्मास्यूटिकल कंपनीज इंडिया में सबसे ज़्यादा हैं। पूरा दुनिया देखती है कि कोविड की दवाई अगर मास में मैन्युफैक्चर होगी तो हिंदुस्तान द्वारा होगी। जो मैंने 6 एरियाज बोले मैडम, हम इसमें भी बहुत ज़्यादा प्रगति कर सकते हैं। और लास्ट में मैं यही बोलना चाहूँगा कि आपका बहुत बहुत धन्यवाद। हम राज्य सरकार से सारे हमारे सारे कॉलेज के स्टेक होल्डर्स बहुत खुश हैं मैडम, बहुत अच्छा काम हो रहा है। और ग्रेटर नोएडा में जैसे मुख्यमंत्री जी ने बोला एयरपोर्ट भी आ रहा है, तो मैडम ये एक हब बनता जा रहा है। तो मेरी आपसे गुजारिश रहेगी एक बायोटेक्नोलॉजी पार्क का सपना जो हम संजोए हुए हैं वो अगर ग्रेटर नोएडा या आसपास के किसी जिले में आए तो बहुत अच्छा रहेगा। बहुत बहुत धन्यवाद और मैं आभार प्रकट करता हूं आपका दोबारा आने के लिए मैडम, बहुत बहुत धन्यवाद। थैंक यू।
थैंक यू सर। इंडीड बायोटेक्नोलॉजी का फ्यूचर इंडिया में बहुत ही ब्राइट है और हमें इसके लिए सबको मिलकर काम करना है। नाउ आई हार्टली वेलकम आवर चीफ गेस्ट ऑफ द डे, Member of Legislative Assembly from Kanpur and Minister of Uttar Pradesh State, Honorable Shrimati Nilima Katiyar, to give us some words of wisdom and shower her blessings on us all. Ma'am, please.
धन्यवाद हिना जी। हिना ही नाम है ना, मैं ठीक बोल रही हूँ?
जी हिना।
नोएडा इंस्टीट्यूट ऑफ इंजीनियरिंग एंड टेक्नोलॉजी, उसका डिपार्टमेंट बायोटेक्नोलॉजी, जिन्होंने एसोसिएट किया है स्वदेशी साइंस मूवमेंट को, तो सबसे पहले तो मेरी बहुत शुभकामनाएं। और मेरे लिए बहुत ये फीलिंग बहुत अच्छी एक आई कि स्वदेशी साइंस मूवमेंट के साथ हमारा जो NIT है उसका बायोटेक डिपार्टमेंट आगे आया है और स्वदेशी साइंस मूवमेंट के साथ अपने आप को जोड़ रहा है। न सिर्फ जोड़ रहा है बल्कि ये पूरा इस कॉन्फ्रेंस के थ्रू नेशनली नहीं इंटरनेशनली लोगों को ज्वाइंट करके हमारे जो रिसर्चर्स हैं, हमारे जो साइंटिस्ट हैं, हमारे यहाँ से जो एक थॉट्स निकल कर आ रहे हैं उनका इंटरचेंज कैसे हो, और जो बहुत हो रहा है, बहुत हो रहा है। और उससे भी बड़ी बात है कि आपदा के समय, इस इतनी बड़ी सदी की सबसे बड़ी वैश्विक आपदा, ऐसे समय पर आप सब ने, पूरे NIT डिपार्टमेंट, पूरे परिवार ने और बायोटेक्नोलॉजी स्पेशली परिवार ने स्वदेशी मूवमेंट के साथ अपने को साइंस के एरिया में जोड़ा है। मेरा बहुत बहुत आप सबको अभिनंदन है इस दिशा में आगे बढ़ने के लिए, सोचने के लिए। और जो पार्टिसिपेंट इस पूरे 2 दिन की इस मैं इसको वर्कशॉप कहूँ, मैं नॉलेज एक्सचेंज कहूँ, मैंने किस टर्म पर ले जाऊँ, 5 सत्र हैं इसके। ये मेरी शुभकामनाएं हैं कि ये 5 सत्र जो उद्घाटन हुआ है, जिस प्रकार से भट्ट जी ने, जिस प्रकार से जो डायरेक्टर हैं उन्होंने, और जिस प्रकार से रमन जी ने अपनी बातों को रखा है, तो पूरी संभावनाएं मुझे 5 सत्र की, और जो प्रवीण सुनेजा जी ने अपनी बातों को रखा है, एक पूरा विजन और थॉट के साथ ये 2 दिवसीय मुझे लगता है कॉन्फ्रेंस शुरू होने जा रही है। तो बेसिक्स क्लियर हैं, निश्चित रूप से उसी प्रकार का पूरा फ्रेमिंग होगा इन 5 सत्रों का। एक पूरा किन किन टॉपिक्स को हेड करके, लीड करके हमें ज़्यादा आगे जाना है, और जो हमारी नए रिसर्चर की पौध है उन तक जो हमारी पुरानी चीज़ें हैं वो किस प्रकार से आगे बढ़कर जाएं, इस पर हम सोचेंगे, बात करेंगे। और निश्चित रूप से कहीं ना कहीं बहुत सारी दिशाओं की उसमें बहुत सारे अन्वेषणाओं के परिणाम की तरफ ये 5 सत्र की कौन सी बात कहाँ से निकलकर किस इनोवेशन को टच कर जाएगी और उसको फ्लो करके आगे ले जाएगी इन 5 सत्रों के अंदर। क्योंकि बहुत सारे पार्टिसिपेंट इसमें ऐसे होंगे जिनके बहुत सारी मानसिक जिज्ञासाएं होंगी, और उनका जो इंटरेक्शन होगा उससे कहीं ना कहीं सबको ही लाभ मिलने वाला है। हम अभी बहुत सारी बातें हम बायोटेक्नोलॉजी के बारे में सुन रहे थे, और मेरे ध्यान में नाम आ रहा था भारतीय वैज्ञानिक डॉक्टर हरगोविंद खुराना जी का, जिन्होंने DNA के उसकी पूरी टेक्निक के ऊपर जो बेस दुनिया को दिया है। और हम जिस तरह से आगे बढ़ रहे हैं, अभी बात हो रही है बायोटेक्नोलॉजी एक पार्क डेवलपमेंट की, और जिस तरह से हमने नए नए इनोवेशन से, और ये हम कर रहे हैं, पहली बार कर रहे हैं ऐसा नहीं है, ये जो हमारी सनातन परंपरा है, भारतीय चिंतन पद्धति है, संस्कृति है, जिसके मूल में वाक्य है कि नित्य नूतन चिर पुरातन। वो हमारा बैक पलट कर आता है और हमें तत्कालीन परिस्थितियों के अंदर वैज्ञानिकता के साथ अपनी पूरे अनुभवों की सामर्थ्य की श्रृंखला को जोड़ते हुए नवोन्मेषी कैसे हो सकते हैं, ये भारतीय परंपरा से, भारतीय जो मनीषा है और भारतीय जो साहित्य है। अब उपनिषद आज जो पूरा विश्व का विज्ञान है वो नित नई खोजें कर रहा है, और उसको जब हम देख रहे हैं कि उससे कहीं आगे हमारे उपनिषदों में उन सब चीजों का उल्लेख है। अभी जो नित नए अन्वेषण हो रहे हैं, उनकी रिसर्च का रिजल्ट निकल कर आ रहा है, उनका बेस लाइन हम कहीं ना कहीं अपने साहित्य में पाते हैं, अपनी प्राचीन ग्रंथों में पाते हैं। आखिर और यही कारण है कि जब नेस्ले और वो आता है तो हमारा अमूल जो है व्हाइट रेवोल्यूशन, वो हमारा अमूल आगे निकल कर आता है। हम कैसे जो पिंगला है हमारा छंद शास्त्र जिसमें विकसित किया छंद शास्त्र के अंदर पिंगला के सिस्टम को, और वही बाइनरी नंबर सिस्टम आज हमारे कंप्यूटर पर पूरे दुनिया के अंदर वो बेसिक एक निकल कर आया है। 22 रिएक्टर हमारे देश के अंदर हैं जो दुनिया के अंदर सबसे ज्यादा सुरक्षित और कम मेंटेनेंस में चलने वाले रिएक्टर्स हैं। जो अभी मेडिकल टूरिज्म की बात हो रही थी कि हम सबसे ज्यादा सेफिएस्ट इस फील्ड में निकल कर आ रहे हैं, और एक साथ 100 से अधिक सैटेलाइट हम प्रक्षेपित कर सकते हैं, और बहुत सारे देश हमारे PSLV से प्रक्षेपित करते हैं अपने सैटेलाइट्स को। ये सब चीजें हैं जो हमारे नए वैज्ञानिक आ रहे हैं, नए थॉट्स लेकर आ रहे हैं। अगर ये उन सारे थॉट्स को पोषित करते हुए उनके अंदर ये भाव भी आए कि वो एक बहुत बहुत समृद्ध रिच हमारी वैज्ञानिक परंपरा को संवाहित करने वाली कड़ी के रूप में है, तो ये जो भान है और ये जो स्वाभिमान है, ये उनकी निश्चित रूप से जो क्षमता है इनोवेशन की, उनकी नई क्रिएटिविटी की जो क्षमता है उसको इनहेंस करेगी। क्योंकि स्व का जो हम स्वदेशी का मूवमेंट है, स्व का जो मान है, उसको फैक्ट्स के साथ, एग्जाम्पल्स के साथ आने वाली पीढ़ियों के सामने रखना और उसको सिद्ध करना। अभी वो यात्रा जो है, पूरी दुनिया उस यात्रा पर निकली हुई है और आगे बढ़ रही है। निश्चित रूप से भारतीय मेधा ने, भारतीय वैज्ञानिकता ने हमेशा जो चुनौती इस क्षेत्र में आई, उस चुनौती का जिस आयाम का प्रतिक्रिया आनी चाहिए थी, जिस क्षेत्र की, उस आयाम की प्रतिक्रिया स्वरूप समाधान के रूप में यहाँ की मेधा विशेष रूप से आई है। हम नहीं भूल सकते 1965 का और युद्ध पाकिस्तान के साथ, और अमेरिका ने हमारा गेहूं रोका, और जब ये गेहूं रोका तो कैसे PL 480 को रोका, और कैसे आज जब हम बात कर रहे हैं तो कई देशों के निर्यातक हम बन चुके हैं। तो ये जो आपदा आई है कोविड, निश्चित रूप से हमारे यहाँ बहुत सारा एक वैज्ञानिकता को उसने चैलेंज किया है, और वैज्ञानिकता ने उसी फेज में उसको प्रतिक्रिया दी है। नए नए इनोवेशंस आ रहे हैं, और हमने जो पहली PPE किट मार्च के अंदर हमने दूसरे देश से मंगाई थी, आज हम कई देशों को निर्यात कर रहे हैं। तो ये भारतीय मेधा है। निश्चित रूप से आपने जो कॉन्फ्रेंस का आयोजन किया है, इस कॉन्फ्रेंस के माध्यम से न सिर्फ इनोवेशन को एक नया नजरिया इंटरेक्शन के जरिए से मिलेगा, आपस में मिलेगा, और जो स्पीकर्स के थ्रू उनको एक नॉलेज जाएगी, उसका भी लाभ हमारे उसको मिलेगा। मैं देख रही हूँ कि नोएडा इंस्टीट्यूट ऑफ इंजीनियरिंग एंड टेक्नोलॉजी, मेरा दूसरी बार जुड़ना आप सबसे हो रहा है, लगातार अपने प्रकार से जो इस वैश्विक आपदा के समय हम फिजिकल डिस्टेंस के रूल को फॉलो कर रहे हैं, इस समय भी अपनी सारी प्रक्रियाओं को प्रयास करके अपने दायित्वों का निर्वहन करने में एक महत्वपूर्ण भूमिका मैं देख रही हूँ वो निर्वहित कर रही है। और पुनः मैं डॉक्टर DP भट्ट जी को, और जो हमारे डायरेक्टर हैं, और हमारी डॉक्टर लक्ष्मी मिश्रा जी, जिन्होंने इस पूरे को ऑर्गेनाइज किया अपने डिपार्टमेंट के थ्रू, और स्वदेशी साइंस से, मैं इसको कहूँगी, ये मूल को पोषित करना है, उस तरफ से जोड़कर अपने इनोवेशंस को आगे बढ़ाने की बात करी है। मेरा बहुत बहुत सराहना, शुभकामनाएं, साधुवाद, अभिनंदन आप सबको और आपकी इस कॉन्फ्रेंस की सफलता के लिए। विशेष रूप से ये इस समय पर हो रही है, बहुत बड़ी बात है और मुझे भी आप सब ने जोड़ा, बहुत बहुत धन्यवाद।
Thank you so much, ma'am, for your words of wisdom and motivation. Now I request Dr. Pankaj Tyagi, who's Dean Research and Development, NIET, to give vote of thanks to all the dignitaries and guests. Sir, please.
Thank you, Ina. Good morning, everyone. It is my privilege to deliver a vote of thanks at the inaugural session of this international conference. I would like to thank our special chief guest, Honorable State Minister Srimati Nilima Katiyar, who despite her busy schedule has found time to grace this occasion. Thank you, ma'am. I also express my sincere thanks to Dr. D.P. Bhatt, President Swadeshi Science Movement, Delhi, for his continuous guidance and valuable contribution. Thank you, D.P. Bhatt, sir. We are grateful to our Executive Vice President, Mr. Raman Batra, sir, for his words of encouragement. His able guidance has always encouraged us. I thank him for this kindness and continued support. Thank you, Raman, sir. I thank to our well-wisher, Director General, Mr. Parveen Sanuja, sir, for his support, vision, and commitment. Thank you, Parveen Sanuja, sir. I thank to Dr. Vinod Kapse, Director of this institute, for their continuous guiding and support for organizing such type of event. Thank you, Dr. Kapse, sir. My heartfelt thanks to the Dr. Lakshmi Mishra, HOD of this department, and my faculty members for this valuable contribution, guidance, and encouragement. I thank the members of the media persons for covering the event and good hearts who worked behind the screen. Last but not least, I thank to all participants joined through online platform from different institutions for your cooperation is making this inaugural session a resounding success. Once again, I thank you all for your attention. Thank you. Hina, you are unmute. Mute. Yeah.
Yeah, thank you, sir. So now I request all the dignitaries here to please switch on their camera and inaugurate the souvenir of the conference. Now I request everybody to please raise at their chair for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंग विंध्य हिमाचल यमुना गंगा उज्ज्वल जलधि तरंग तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता
Thank you, everybody. So, thank you for a wonderful session. Now, next session will continue after lunch. Please stay tuned.
Good afternoon, everyone. I am Meenu Agarwal, Assistant Professor in Biotechnology Department. Welcome you all for joining us today for two-day International Conference on Biotrends, organized by Department of Biotechnology, NIET. It's my pleasure to introduce our first speaker for the day, Dr. N.G. Lakshminarayan. Dr. N.G. Lakshminarayan, he is currently the Chief Business Development Officer, National Research Development Corporation, New Delhi. Dr. Lakshminarayan has commercialized the innovations and technologies developed by the laboratories associated with the Central Silk Board and other R&D organizations. In the past, Dr. Lakshmi Narayan has headed as Senior Manager, Business Development and Coordinator, IFPC, NRDC, Bangalore. He has also marketed technologies and concluded 237 technology transfer license agreements and generated revenues of more than rupees 4.22 crore from technology transfer to his credit. He has successfully implemented intellectual property facilitation center project funded by Ministry of MSME, Government of India and NRDC at Bangalore. Dr. Lakshmi Narayan has established a rural technology demonstration cum training center in South India and an insect pathology laboratory at UAS Bangalore. Dr. Lakshmi Narayan has also collaborated with R&D organizations, universities, institutes, academia, NGOs, and industry associations for providing IP protection and technology commercialization services. We are pleased to have you here, sir. Please enlighten us with your thoughts. Sir, please switch on your camera. Sir, please switch on your camera. Sir, please unmute.
The camera is on?
Yes, sir.
Camera is on now? Camera is on. Camera is on. Can you see?
Yes, sir. So please start.
Okay, thank you very much for giving me this opportunity. I thank Dr. Rashmi Mishra. Just a minute. Madam, बोलिए. कैमरा दोनों ऑन है मैडम. On. Oh, is it? But the presentation is on. Presentation.
Yes, sir. Presentation is visible.
I'm able to hear my voice. Yeah, can I? Am I audible now? Am I audible now?
Yes, sir.
Oh, it is very slow. Network is slow. Just give me. network is slow, but I don't know whether you're able to hear me.
Yes, sir. We are able to hear you.
Okay. Thank you very much, Dr. Lakshmi Mishra, as well as Dr. DP Bhatt for giving me this introduction to Noida Institute. I would have loved to visit your place and give this talk. Amid this pandemic, we are meeting this online. Oh, the network is very slow. Just give me a moment. What? But it is. Hello.
Yes, sir. You are audible, sir.
Can I go on now?
Yes, sir. Please continue, sir.
Okay. Participants, I think majority of you are from the life science background, and now you see there is a lot of opportunity for people who have started this uh, the the background in which you are looking for. You have, I think, entered into bachelor's or master's programs, and you would be doing a lot of research. What is it that you do in research? You are aiming at finding something new in the research, right? And when you do research, what is your objective? Your objective is to find something new. There is a lot of I'm switching off the camera now. The network is very slow. Kindly share the presentation only, please. Well, let me briefly tell you what our organization aims for once you do the research. The organization in which I work is called National Research Development Corporation. We are basically a public sector enterprise under the Ministry of Science and Technology, which is catering to the needs of the R&D as well as to the industry. Whatever development is happening in the R&D is taken forward to the industries through NRDC. our basic objective is to take innovations to the industries, and in the process we also provide services for protecting, developing and commercializing the technologies. So I'll quickly go to the next slide. See, you can see the strength of NRDC here, our expertise, our partnerships and achievements over the years. We are basically a technology transfer organization wherein we process technologies or research accomplishments of R&D for their future potential in industry. We have strong linkages with the R&D as well as the industry across the country, and we have concluded almost about 5,000 technology license agreements as of now. And before we actually start talking about what these technologies are, so basically when industry approaches us, the industry wants technologies that are protected. So knowledge that is protected has value. You are all aware about it. So what is the kind of protection available for the knowledge or the research achievements of the R&D? So there are several tools that are available, the legal tools as well as the secrecy tools. Here you are seeing here. The main important tool for protection of technologies are patent, copyright, trademark, design registration, geographical indications, layout design of integrated circuits, plant varieties. These are the tools that are available for protecting the knowledge that is created in the R&D. So I will not go much into detail. Patent is very important. As you are aware, the functional features of any new development that is taken as it is developed is usually protected in the form of a patent. So what are the kind of tools that you need to use? We will advise the R&D institutions how to go about when they enter into some kind of a memorandum of understanding with NRDC. We will guide the institution and the scientists on how these tools have to be utilized for protecting their knowledge that has been developed over the years. Which tool to be used? I don't think there is any super hit formula to for this. Usually, we always combine tools that are used for protecting knowledges that are being developed. Often they are used in combination, and when it is used in combination, we usually call it as a technical know-how with a bundle of IP rights. So this is the NRDC operation model. On the left, you see the technology sources. NIT also, and Noida Institute also could be one of the sources of technology. And these are the value additions that NRDC does for the technologies that are developed by the institutions. And on the right, we see the industries, entrepreneurs, startups to whom we advance these technologies or the users of the technology. These are the various services that we offer. The process of development of technology. Basic information that is provided by the R&D institutions are evaluated techno-commercially. Once this is evaluated and we get a positive feedback on the technology level of adoption, we go into licensing activities. Parallelly, we also provide some kind of a financial support to the R&D institutions for proving their prototypes or doing some kind of a field trials or for product testing or any such small activities in which there is some kind of a value addition in terms of the experimentation that is being done at the R&D institution. Additionally, we also do market survey for finding out what is the potential of the technology. We also carry out basic engineering design package studies, and we also carry out feasibility studies. Many other studies we do at our cost before we actually offer this technology to the industry for licensing. And wherever possible, the technology is successfully commercialized by industry in the country. And if there is a demand for such a technology elsewhere outside the country, so we export these technologies on a turnkey basis. And we also have a dedicated technology portal on our website where the information on technology seekers as well as technology offers that the industries, the R&D develops can be placed on the portal, and the information could be exchanged. We also carry out detailed project report studies, diagnostic studies. We have a lot of technology database on technologies as well as patent databases. Nowadays, patent databases has become online, and we have dedicated subscription tools for doing patent search, analysis, and a lot of other studies which are required for industry to get the confidence in launching products or entering into a new market.
So, sorry to interrupt you, sir. Sir, your slides are not moving. So please scroll down. Sir, your slides are not moving.
Slides are not coming.
No, sir. So please scroll down. We are not able to see the slides.
You can see it now.
Yes, sir. Thank you, sir.
You can see that?
Yes, sir. Thank you, sir.
Shall I expand it?
Yes,
now?
Sir. Please continue. Yes, sir.
Yeah. Oh. What about? Just a moment, I'm just changing. नहीं अभी मैं इसमें कॉन्फ्रेंस में Can you see the slides now?
Yes, sir.
Okay, I was talking about the services that NRDC offers. So these are the main services or the mandatory services that NRDC offers. in technology evaluation, what we do is when an R&D submits its proposals for commercializing NRDC to technologies through NRDC, we basically do a techno-commercial evaluation. So what we look into that is the techno-commercial feasibility of the technology for its adoption in the industry. Hello, can you hear me?
Yes, sir.
Yes, sir.
Okay. In addition to that, we also provide a small kind of support to the R&D institutions for carrying out some kind of a prototype development or a field trial or a
Sir, your PPT is not visible, sir. So I would like to request you to please make your PPT visible, sir.
Yeah, the call has dropped. I'll just share it again. You're able to see now?
Yes, sir, now it is visible. You may proceed, sir.
In addition to that, we also carry out market surveys, basic engineering design, feasibility studies. We prepare DPRs for projects for the industries who are interested in getting these done. And in addition to that, we also provide access to technology data bank and patent. Yeah. I have shared it.
Yes, sir, PPT is visible, sir. You may
Okay.
proceed, sir.
It's visible now.
Yes, sir, it's fully visible. You may proceed, sir.
Okay. We also provide support to our parent ministry in pushing their schemes. Okay. Okay. In the area of promoting technologies, we also give away awards on technologies that are available for commercialization. We give away awards meritorious inventions every year in the form of a national prize award, national meritorious invention award. These are the different kinds of awards that are being offered to the R&D institutions who develop new technologies. We also participate in a lot of exhibitions showcasing technologies. We also publish magazines in Hindi as well as in English. Hindi, we call it Avishkar, which is published on a monthly basis. And Invention Intelligence is a magazine which we publish bi-monthly basis. And we also carry out a lot of interaction meets with association of the industries and also academic institutions to reach out to industries who are interested in acquiring technologies from NRDs. And in addition to that, we also provide consultancy services for protecting IP and we also provide services for doing patent search, as I was telling you earlier. We conduct a lot of awareness programs, seminars, workshops, and we also make policy for IP policy for academic institutions, R&D institutions, industries. These are some of the network of institutions with whom we have been associated. Name them CSIR, ICR, ICMR, some of the big universities in the country, the state agriculture universities, and a lot of institutions with whom we have been associated. And this is just a partial list. In fact, even corporates like Navali Lignite Corporation also has been associated with NRDC for transferring some of their technologies which they have developed. These are some of the customers and partners to whom services have been provided. There could be some of them could be licensees, or some of them could be corporates who have taken some kind of a service like patent protection or patent prosecution from NRDC. As I was telling you about the prize award categories here, we have network problem. हाँ, क्या नेटवर्क 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 मैंने अपना भी डोंगल भी लगाया और ये ऑफिस का भी लगाया, दोनों में भी ऐसी है। ठीक है। I think there is a network issue here, and I'll wind up my talk here. And we will definitely meet some other time and personally visit your institution. I would like to visit institution and maybe carry forward a few things that NRDC can offer, and we will exchange information about whatever technologies have been developed by your institution to take it forward either for protection or for commercialization in the future. Thank you very much, participants. I'm very sorry there is a network issue. We will definitely meet sometime on a one-to-one basis. Sorry for the trouble that the network has been giving us today. I'll close the presentation here. Thank you very much for giving me this opportunity. We'll definitely meet again sometime very soon.
I'm Dr. Pratibha Pandey, Assistant Professor in the Department of Biotechnology, and I would like to thank Dr. N.G. Lakshmi Narayan sir for his insightful presentation. Your lecture was highly informative and interesting, sir. Definitely, it will help our students as well as our participants to further enhance their, or you can say widen their skill and technology in the area of research and development. This will definitely help them to further explore the area of patent, copyright, so that they can also save, not only invent, but also know the ways to safeguard their invention. Thank you so much, sir, for your insightful presentation.
Ma'am, I request you to please unmute yourself. Now I'd like to introduce our next speaker, Dr. Puneet Gandhi. Professor Puneet Gandhi did her PhD with the prestigious K.S. Krishnan Fellowship from Baba Atomic Research Center, Mumbai. She is the recipient of the ISCA Young Scientist Award in the medical section. About a decade earlier, she specialized in hospital and healthcare management from Symbiosis, Pune. Since then, she is engaged at Bhopal Memorial Hospital and Research Center. She is currently working in the area of translational research, especially on non-invasive biomarkers in non-communicable diseases. Dr. Gandhi has an experience of clinical research of more than 30 years and has completed more than 12 extramural national level projects and 100 pilot projects in the last 10 years. She is also a coordinator for skill upgradation program in medical biotechnology for the State Biotechnology Council and DBT. Dr. Puneet Gandhi has been teaching in various capacities since the past 18 years and has 39 international publications and 60 paper presentations to her credit. She is a mentor for PhD in medical biotechnology for various universities and has PhD students registered to work on molecular detection and disease of non-communicable diseases. Ma'am, we welcome you and request you to share your thoughts with us. Ma'am, I request you to please on your mic and camera. Ma'am, I request you to please on your mic and camera. Now, I'd like to introduce our next speaker, Dr. Lakshmi Narayan Mishra. Dr. Lakshmi Narayan Mishra is currently a postdoctoral scientist at the Albert Einstein College of Medicine, New York, USA. He was awarded PhD in chromatin biology under the guidance of Professor M.R.S. Rao, former President, Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore. His work focuses on understanding the role of linker histones in regulating the gene expression. He has published several research articles in international peer-reviewed journals of high repute, including in the Proceedings of the National Academy of Science, USA, Journal of Biological Chemistry, USA, Nature Scientific Reports, Epigenetics and Chromatin, Biochemical Journal, Journal of Proteomics, etc. For his extraordinary contribution in the field of epigenetics and chromatin, he has been awarded Harry Eagle Scholarship for the Outstanding Postdoctoral Fellows by Albert Einstein College of Medicine, New York, USA. He is a recipient of several international scholarships and travel grants. He is an early career reviewer, board member of the Journal of Biological Chemistry, USA, and is also serving as the associate editor of the Bioprotocols, an international peer-reviewed journal published by the Research of Stanford University, USA. Now, I'd like to invite you, sir, to please enlighten us with your thoughts.
Hello. Are my slides visible? Hello.
Yes, sir. No, sir, your slides are not visible, sir.
Okay, so I think there is some issue with the sharing. Hello.
Yes, sir. Sir, you're audible, sir.
Okay, so can I send you the presentation in email and can you open share from your side?
Oh, okay, sir. Sure, sir.
I'm just sharing it.
NIET.
Yeah, just sent it.
Sir,
So it went to Rasmi ma'am's account, Gmail
okay,
account.
sir. Okay, sir.
Yeah. Did you get my email? Hello.
Yes, sir. We have received your mail, sir.
Okay, good. Let us start sharing from there. Can we start now?
Sir, just 1 minute. We are opening the PPT.
Okay. Yeah, please go in presentation mode.
Please continue, sir.
Yeah, please share the screen, whole screen, so that so that people can see it. Yeah. Okay, so thank you for the nice introduction and thank you for inviting me to be one speaker in this international conference. So today I'm going to talk about linker histones, and I would try to cover it from my PhD to my postdoctoral journey that I am doing still now. So this is the overview of my presentation. So first I will introduce all of you to the chromatin, and then I will discuss about the functions of linker histone in spermiogenesis. That is one process when by which sperms are formed. And then I will go on discussing about the role of linker histones in controlling the chromatin accessibility. Role of linker histones in modulating core histone PTMs, and then I will summarize, and then we will have some question answer session. So what is DNA? How DNA is packed inside the nucleus? So all of us know that DNA is the genetic material, meaning it carries all the information that is needed to form a body, all the system processes that happens in the body, everything is there encoded inside the DNA. And it has only 4 bases, A, T, G, and C. So adenine, thymine, guanine, and cytosine. If you see the total length of DNA in each cell, it is almost like 2 meters long. However, it has to fit inside the nucleus that has the diameter of only 10 microns. So how is it possible that you can fit 2-meter long DNA in each cell in 10 micron diameter nucleus. So it's not feasible in general case. So there are several types of proteins called histone proteins and non-histone proteins that help in this fitting process. So these histone proteins are of 5 types: H2A, H2B, H3, and H4. We also call them as core histones. And there are linker histones. They are 11 subtypes in mammals, and they are called linker histone H1s. So this gene expression depends on how chromatin structure changes with time or when they get any stimulus. So this chromatin accessibility plays a major role in gene expression. So what is gene expression? I will come later on. So if we see on the right side the structure of DNA, so this is a DNA, and when it has to fit inside the nucleus, it cannot fit alone. So it has to bind to the histone proteins. You can see here on number 3. So this association of histone protein with DNA is the structure which is formed by their association is called nucleosome, and nucleosome are also called the structural and functional unit of chromatin. So DNA inside the nucleus is present in the form of chromatin, and the structural unit of chromatin is called nucleosome. So we have to remember this throughout my presentation. So there is a 5th kind of histone that I mentioned, H1. This H1 is found outside the nucleosome, and it is bound to the linker DNA. So what is linker DNA? Linker DNA binds 2 nucleosomes together. So linker DNA binds 2 nucleosomes together, and linker histone, the name as it suggests, it is found in the linker DNA, and it helps in the folding of these nucleosomes on each other. So H1 is essential for chromatin condensation. It condenses. It is no longer like it's not longer open. It is getting closed when H1 is bound to the nucleosomes, and these structures further condense with the help of nucleosome histone tail interactions. So all these H2A, H2B, H3, and H4 have N-terminal tails that is protruding outside the nucleosome, and they interact with each other and help in the formation of condensed chromatin, and H1 adds to that condensation. And this is the mechanism by which all the DNA packs inside a tiny nucleus. So how DNA passes from parents to offspring? As I mentioned, that all the information is inside the DNA. So this has to pass from generation to generation. Whatever I have got the information that has come from either from my mother or my father. So what carries all the information? And the answer is DNA. So DNA is passed through the sperm that goes in and mixes in the form of pronuclei, sperm pronuclei with the egg pronuclei, and fertilization, it forms a zygote, and zygote has all the information. It's a totipotent cell that can form all the body part of the mammalian system or any living system that has sexual reproduction. And then offspring are formed, and these offsprings had DNA that has come from either the father or the mother. So this DNA has the capacity to form all the body parts. So how is it possible? There were only 2 cells. Sperm is haploid cell, egg is haploid cell. When they mix together, means fertilization happens, it forms a diploid cell, that is zygote. And then zygote has all the memory. So what is there to give the memory? So there are, for the memory, there are histones that has been retained during this process, a spermatogenesis process that I will discuss. That is my research actually. And ovum also gives some information about what to form, which body part to form. So everything is there. All the information is there in the zygote. So then comes the flow of information, how the organs will be formed. So this is called central dogma in molecular biology. Means DNA forms RNA by the process called transcription, and RNA forms protein by the process of translation. And this process is called gene expression. Means DNA is giving a functional product either in the form of RNA or in the form of protein. And since DNA, as I mentioned, is bound to histone proteins or non-histone proteins or even RNA, they are inaccessible to any other transcription factors or any other transacting factors. No transacting factor can bind to DNA if it is bound to histone protein or non-histone protein that has much more affinity. So no one wants to leave their friend, and DNA and histones are friends. So these DNA are not accessible. So to make them accessible, histone has to go away. Otherwise, there won't be any friendship with other transacting factors. So chromatin accessibility plays a major role in transcription process. So how will you change the chromatin accessibility? So there are three different mechanisms by which you can change the chromatin structure. First is chromatin composition. So as I mentioned that nucleosome is the structural and functional unit of chromatin. So you change the nucleosome. So how will you change the nucleosome? So as I mentioned that there are 5 histones, 5 types of histone, H2A, H2B, H3 and H4 and H1. So you change those histones, canonical histones with variant histones. There are several variant histones for H1. I mentioned 11 histone variants are there for linker. There are no linker and there are no variants for histone H4, but H2A has many histone variants. So all the histone variant have different characteristics. Although they look similar, there are 3 or 4 amino acid changes, but their structure and function are completely different. So 1 or 2 amino acids can change everything inside the nucleus. That's by mutation or substitution, anything can happen and function is going to change. So we change the composition or we modify them. So histones can be modified by post-translational modifications, and those post-translational modifications are covalent modifications such as acetylation, methylation, phosphorylation, sumoylation. There are umpteen number of modifications that can happen, and they can change the structure and function of the histone, thereby changing the accessibility also of the chromatin structure. And third is by changing the position of the regulatory elements. So for example, regulatory element is here and it is bound to the histone. No other protein can come and bind to these regulatory elements. So there are modifiers called ATP dependent chromatin remodeling factors which can remodel them. So how they will remodel? They will remodel in such a way that these nucleosomes can slide little bit. They start moving with the help of ATP because ATP is the energy source there, and these will be, after some time, these blue region inside the nucleosome will be accessible and regulatory elements will come and bind, regulatory factors can come and bind here. So these are 3 mechanisms which I will be talking about today. So as I mentioned, there are several H1 variants or linker histone variants. These are of 2 types, somatic and tissue specific. And tissue specific histone variant was my project during PhD at JNCSR Bangalore. These tissue specific histone variants are of again 2 types because there are 2 types of tissue based on the mammalian sex. So these are testis specific or oocyte specific. So oocyte specific are only found in female. Testis specific are found only in males in the testes. So testis specific are again of 3 types: H1T, H1T2 and HILS1. HILS1 was the newest member when I joined my PhD program in 2006, and this protein was reported only in 2003 with no known functions of this protein. So these were the properties that were known at that time when I joined the PhD program. Only the first 3 facts about this protein. So this is a 19.2 kilodalton protein, expresses in elongating and elongated spermatids. So these are the steps, and these are the shapes of the spermatid during maturation into mature form. Interacts with transition protein and protamine. I will discuss about this in next slide. And then someone in 2007 lab showed that this protein has significantly lower level of HILS1, has lower level of transcript, and that was reported in infertile male that had asthenozoospermic condition, means motility was very less in those males, sperm motility, and sperm motility plays a major role in fertility. And transition protein 1 and HLS1 double knockout mice were infertile, means there is some property in this protein that plays a role in fertility. And when you go to a doctor and doctor says that, okay, yeah, everything is fine, there is no problem with the male or the female, but the female is not getting pregnant. What is the rule? What is the reason behind that? So nobody knows that what are different other proteins which are playing a role in that spermiogenesis process. And that was the purpose of studying this protein. As a biotechnologist, so when I joined biotechnology program, I was told that biotechnology is application of technology for the betterment of human life. So whenever we study any protein, any RNA, any DNA, we look for some application. So I found that there is application of this protein in spermiogenesis or fertility. So HLS1, as I mentioned, it expresses during elongating and elongated spermatid, and that is also the step. These are the elongating and elongated spermatids, and these are also the steps when chromatin remodeling takes place. So what is chromatin remodeling? So chromatin remodeling is when most of the histones that we know, that I mentioned also, that histones are bound to DNA, but there are exceptions. Sperm is one exception where most of them, 95% of the histones are replaced first with transition proteins. These are called transition protein 1 and 2, and they are further replaced with more basic protein called protamines. So what is the necessity of these histones are there in all of the body cells? Why a sperm has different protein? Because this sperm has to travel inside the female reproductive tract, and the female reproductive tract has a pH of 3.5 to 4. It is acidic. A sperm can get damaged if it is not condensed, highly condensed. So it has to condense to spare itself from DNA damaging agents. So if you read about the sperm or ejaculated sperm, you will see that the pH is alkaline, slightly alkaline, and that is because a sperm within 30 minutes it will be neutralized, and the female cannot get pregnant because at least one sperm has to go and meet with the egg. And there are like 50 million or 60 million sperm, but to the surprise, only one sperm meets with the ovum. So, and if it does not, even one cannot, it is called infertility. So since this protein plays a role in infertility, it is worthwhile we should know about infertility. What is infertility? So infertility is inability to achieve pregnancy even after 1 year of having regular unprotected intercourse, or after 6 months if the woman is older than 35 years of age. Most, like out of 10, 1 or 2 people are suffering for infertility. Nobody tells, but it's the reality. It affects an estimated 15% of couples globally. It's everywhere, in US, in India, in Japan, in China, everywhere. Many different medical conditions and other factors can contribute to fertility problems, and every individual may have single cause or multiple cause on infertility, and there may be some unidentifiable cause also. For example, if the protein level is low, doctor cannot tell that this protein level is low because it's very costly business for them also, and there are no like studies showing that, okay, you can study the expression of this protein. You will have to take a sperm in different spermatocytes in different phases of maturation, and nobody wants to give the sperm in different phases because you have to cut the testes. It's very difficult to do that. In 1/3 of the infertile couples, the problem is with man. 1/3 of the infertile couples, the problem is with the woman. And in 1/3 of the infertile couples, the problem cannot be identified or is with both the man and the woman. So in our society, we used to know that, okay, female is the culprit, she cannot get pregnant. But remember, it's equal. equal chance of infertility in man and woman. So possible cause of female infertility is failure to ovulate, accounts for almost 40% of female infertility. This is the most prominent cause, that female is not ovulating. Number of ovums are limited in females. Problems in the menstrual cycle, structural problems of the reproductive system such as fallopian tube and uterus problem, infections such as untreated gonorrhea and chlamydia, implantation failure means embryo is there but it's not getting implanted inside the uterus. Endometriosis means this is endometrium is the lining of the uterus that gets shredded with time if there is some problem. Polycystic ovarian syndrome, if I know correctly, it's like out of 10, 3 women facing this problem, polycystic ovarian syndrome, primary ovary insufficiency, uterine fibroids, and autoimmune disorders. If you come to the male infertility side, there are abnormal sperm production or function due to the undescended testicles. Testicles has to descend from the body because it needs 2 degrees Celsius less temperature from the compared to the body. We have 37 and a sperm matures at 35 or 36 maximum. It cannot mature at 37 degrees Celsius. Genetic defects, health problems such as diabetes or infections such as chlamydia, gonorrhea, mumps or HIV. So please everybody take care of your health. Don't be diabetic. It causes infertility in future. Enlarged veins in the testes, blood supply changes completely, problems with the delivery of a sperm such as premature ejaculation. This is most prominent cause of infertility. Certain genetic diseases, structural problems such as blockage in the testicles. You go, you have a sperm, but it is not coming in the serum, so that's a problem. Or damage or injury to the reproductive organs. Pesticides, radiation, cigarette smoking, alcohol, marijuana, antihypertensives, and anabolic steroids that people take very usually. They're all causes of male infertility. Damage related to cancer and its treatment, including radiation and chemotherapy, they all cause infertility. So infertility, as I told, is everywhere, and these are the males contributing to infertility worldwide. In Asia, we have 30% males, 37% of all the infertile couples, 37% are males. In US, you can see that in North America, it's 50%, it's much more. In Middle East, it's even 60 to 70%. So it keeps on changing. So finally, so fertilization. So if the male is fertile, woman is fertile. So what will happen is fertilization is the process where ovum, that is female gamete, and male gamete is sperm, they meet together and they form zygote, and only one sperm goes inside the ovum, and this is a sperm ovum nucleus, and this is the nucleus of the sperm, and only the nucleus part goes from the sperm, not all the parts. So this nucleus is highly condensed in this, and they have 95% transition protamines, and only 5% are histones that we call residual histones. So my protein comes into play now. So HLS1, as I mentioned, that it was reported in elongating and elongated spermatids. So the first thing, because the properties were not known, so we have to correlate with some property. Someone takes birth in our family. What we do is, my mother comes or my father comes, and they tell that, okay, this looks like this looks like mother, or this looks like father. So in biology also, when we do research, we go towards the family. So family business is the main thing everywhere. You have to correlate. So HLS1, I wanted to see how much similar this is with other linker histones which has been reported in the literature. So all the linker histones have tripartite structure, means 3 parts are there. N-terminal histones that has no structure, globular domain that has a structure, and C-terminal domain that does not have a structure. And most of the functions are done by C-terminal domain of the linker histones. They condense the chromatin, and remember, the main property of a linker histone is to condense the chromatin. So what I did is, I first assembled the sequence of rat H1D. So human is not possible to study for all these types of work. So you have to take some animal, right? So either mouse or human, rat. Those are very similar. They are physiologically similar to human. So I took rat. The reason was we get much more like bigger testes compared to mouse. Mouse is very small animal compared to rat. So rat I took for my study, and my lab in IISc Bangalore, Professor M.R.S. Rao's lab, that has worked extensively with rat H1D and H1T. So we had experience of working with these proteins, except with HILS1 and H1T2. So first thing I did was I assembled their sequence and found that, oh, there are differences in many of the regions. This is N-terminal domain, red region is the globular domain, and again there is C-terminal domain, and in the C-terminal domain, SP or TPKK motif plays a role in chromatin condensation. So TPKK, if you see this TPKK, all the TPKK or SPKK motifs are completely different in HILS1. If you see the sequence, this is completely different. So one thing I knew that, okay, based on the literature search and my background analysis, it cannot be a strong condenser of chromatin, because chromatin condensation motifs are completely different in this protein. And second is DNA binding protein. If it does not bind to DNA, how can it condense the chromatin? So for DNA binding, globular domain is responsible. And in the globular domain also, we could see that 3 amino acids have changed out of 7 amino acids. So it will have altered DNA binding capacity. So the main thing that people want to know about the linker histone property is chromatin condensation property. And as I mentioned, the C-terminal domain was completely different. So I studied DNA binding property. I studied DNA condensation property. I studied chromatin condensation property, and I'm showing here the chromatin condensation part. So what I did is I took the testes, I isolated native chromatin, and I stripped all H1s from there. So there is no H1 now. And then I added back H1D, H1T, and HILS1. So why did I take H1D and H1T? These are reference for me, because these are working as a reference or control for me, because H1D is a strong condenser of chromatin, H1T is a poor condenser of chromatin, and HILS1 property we don't know. So either it will be a poor condenser of chromatin, its property will be similar to H1T. If it is a strong condenser of chromatin, its property will be similar to H1D. So these are serving as a control here. And then I analyze chromatin condensation by circular dichroism spectroscopy. And here you see it's a simple view of the result that I got. So if these are molar ellipticity on y-axis, and x-axis are different chromatins. So native chromatin is much more condensed, and to understand this figure, you have to remember that the molar ellipticity is less, means chromatin is more condensed. If the molar ellipticity is more, it is less condensed. So as you see, native chromatin, where H1 is there, all the H1s are there in the testis chromatin that is native, it is more condensed because the molar ellipticity is less. As soon as I strip H1 is not there, means there is no protein to condense them, the ellipticity goes high, means it is less condensed. When I add 1 H1D per nucleosome, this is molar ratio, it comes back, means it condenses again. So H1D is a strong condenser of chromatin because it is similar to native chromatin, a molar ellipticity. But when I took out H1T, when I took out H1T, when I took out H1, molar ellipticity increased, means it was poor, it was not condensed. But when I added H1T, it was further like less condensed, it was like more open. And similar was the case with HILS1. So we found that HILS1, hello, is my time over? Hello? Hello? Okay. So HILS1 is a poor condenser of chromatin. So the next thing was, okay, so as I mentioned that histone post-translational modifications also play a role in these processes. So I wanted to know how, like, what are the modifications in these proteins. So I separated the testis histones by RP-HPLC. And this is the here is the peak for HILS1. And this study we published in Journal of Proteomics, and we found that there are several residues which undergoes post-translational modifications. These modifications are known in other proteins, not in this, but these were known. So another modification that was serendipity for me, I was just testing several antibodies, and I found that tyrosine and phosphotyrosine antibody also reacted with HILS1 protein and other linker histones also. And this was surprising that how tyrosine phosphorylation is happening in linker histones when there is no report like this. So this was the first report that linker histones undergoes tyrosine phosphorylation, and as a control for that, I had used phosphorylated tyrosine BSA, and as a negative control, I used BSA that has no phosphorylation, and this confirmed this. This was further confirmed by tyrosine a specific phosphotyrosine a specific HILS1 antibody. So then the question is, where does it bind in the genome? You have to know the location of this protein. So HILS1, we did chip sequencing, and chip sequencing showed that it binds primarily with the intergenic and intronic regions of the genome. So why it binds to intergenic and intronic regions? So I looked into the literature and I found that most of the histones, that 5% residual histones, are there where intergenic and intronic regions are present. So those 5% regions which has residual proteins, histone proteins, and that will give signal for the body development when fertilization happens and like child gets birth, takes birth. So throughout the life, starting from the fertilization up to 9 months in the as a fetus, and then throughout the life, there are histones which keeps on giving the signal that okay, this you have to do. This information has come from your mother or your father. So these are the regions which we found that okay, these associates with this protein. So summary of this part is HILS1 is a poor condenser of chromatin. It gets post-translationally modified by phosphorylation, acetylation, and methylation, and it associates primarily with repetitive DNA sequences. These are DNase1 hypersensitive sites and retained parental histones. So coming to the second part of my talk, that is a study of the effect of linker histones on chromatin accessibility. So nobody knew that how much chromatin condensation it can provide, linker histones can provide. So can we reconstitute nucleosomes in vitro? Nucleosomes are present in the body, but can we do outside the body? And the answer is yes. So I joined Professor Jeffrey Hess lab at University of Rochester, who is a world leader in reconstituting this system outside the body. And when we bind nucleosome positioning sequences, these are DNA with histone octamer, so two molecules of each of histone H2A, H2B, H3, and H4, it forms a bits on a string kind of a structure that we all must have studied. You also must have studied in any molecular biology book. So this is a bits on a string kind of a structure, and when we add 0.5 millimolar magnesium, it starts folding, and when we add H1 linker histone to that, it further folds, and at 10 millimolar magnesium, it is highly condensed in nature. So these are this is the process which I described above. So how can we study chromatin accessibility? There are no method. So what I did after discussion with Jeff is I ligated 12-mer nucleosome array. So there are 12 nucleosomes in one system, and there is a central nucleosome which has DRA3 site on both the sides. And then I ligated these 12-mer nucleosome arrays from both the sides that also has DRA3 enzyme, restriction endonuclease site. So when we ligate together, it forms a continuous 25-mer array. And if I bind linker histone here and digest with this similar enzyme, DRA3 enzyme, if it is highly condensed, DRA3 cannot bind there. And the digestion rate will be very slow. If it is open, the digestion rate will be very high. So I did that, and as a control, everywhere in the biological system, you have to have one control. So as a control, I took control DNA, that is naked DNA, nothing. So because this was the linker DNA here, linker DNA we are digesting, so we have to take some DNA that is free of histones. So I took control DNA, and then this strategy we published in Nature Scientific Reports in 2016. So this is the strategy on the right side. what we did with this to study the chromatin accessibility, and we found that in 10 millimolar magnesium, when the chromatin is highly condensed, you can see here this structure, tertiary structure here, fully condensed, and in 0.5 millimolar magnesium, structure is like this, zigzag. So when we digested that, relative rate was almost like 280 times less when wild type chromatin was having H1 bound. So chromatin was not accessible at all. It was 280 times less. And in 0.5 millimolar magnesium, when the nucleosome had folded, it was still 50 times, almost 50 times less accessible. It means linker histone drastically decreases the linker DNA accessibility in partially or fully condensed array. And when there has to be gene expression, linker histone has to come out, otherwise there cannot be gene expression. So this we published in Journal of Biological Chemistry in 2018. So the last part is 2 slides more, what linker histone does more inside the body. So linker histones stimulate the activity of SUB39 enzyme and ZDB1 methyl transferases. So we reconstituted, when I joined the lab, I reconstituted dinucleosome, not 12, not 1, but this time I did dinucleosome, and I added or did not add the H1 to them. So when we added, you can see the red region here, this is H1 added. And then I did methyl transferase assay, means I added SUB39 H1 enzyme, ZDB1 enzyme, and G9A enzyme, and allowed it to bind, methylate the histone H3, and they are known to bind and methylate H3K9, and they produce H3K9 dimethyl or trimethylation. And G9A is found in the euchromatic region. So there are 2 types of chromatin: euchromatin, that is getting expressed; heterochromatin, that is not getting expressed, means transcription won't be there. Euchromatin, transcription will be there. So G9A is found in the euchromatic region, and its activity was not affected. You can see that it's like the intensity of the band is same. But if you compare here, intensity of the band of H3K9 dimethylation or trimethylation was much more compared to when there was no H1. If you compare first lane with second lane, it means linker histone stimulates the activity of SUB39 and ZDB1. So this we published in Proceedings of the National Academy of Sciences this year. And then we also created embryonic stem cells that has very low level of H1 expressing. So this is very low level of H1 expressing, and these are measured satellites, and measured satellites should not express, but it was expressing because there was no H1. So expression was really like happening there. But when we added full length H1, black region, you can see the expression decreased immediately. Means H1 did not allow them to express, and that was the reason that I was studying all these things. And when we added H1D del 75, means del 75 means from the C-terminal domain, we have deleted 75 amino acids of the C-terminal domain. And if you remember, it's very difficult to remember, although it's new topic for many of you. So C-terminal domain was responsible for chromatin condensation. So there is no chromatin condensation, and so the expression will be more. So expression, it was here, expression was very less, it's much more now. It's more than 5 times. It has decreased 10 times, and now it has increased 5 times again. So this we published in PNAS this year. So the last slide is here. So this in this study we wanted to see what are other methyl transferases that gets stimulated or inhibited by linker histones. So this time we wanted to see the PRC2 activity, and if you see H1D, and this is delta CTD, means there is no CTD, we have clipped it off, and this is checking the activity of PRC2. As a control, loading control, we have H4 protein antibody. So Western blot was done with either H3K27 trimethylation antibody or H4 antibody. H3K27 trimethyl, because this protein PRC2 methylates H3K histone H3 at 27th residue, that is lysine, and it gives 3 methyl groups there. So without H1, you can see the band is bent. As soon as I add H1D, band is very intense, means it is stimulating the activity of the PRC2. But when we have del CTD, means CTD is not there, you can see that it's going faint, but not as faint as minus H1. So del CTD is responsible, but not it is not solely responsible for this active stimulation of PRC2 activity. And you can see the graph here again. So summary of my talk today is linker histones regulate the gene expression by restricting the accessibility of chromatin. Linker histones control the gene expression by chromatin condensation. Linker histones C-terminal domain play an important role in chromatin condensation. And linker histones control the gene expression by regulating the core histone post-translational modification. 2 of them we saw, many of them we have studied. But we have not published them. 2 of them we have published. So one is coming in Nature, it's in press. These modifications are usually associated with heterochromatin. With this, I would like to acknowledge my PhD supervisor, Professor M.R.S. Rao, my postdoc supervisor, Professor Jeffrey Hayes from University of Rochester Medical Center, and my current supervisor, Professor Arthur Spalchi. He's very friendly. You can see my son shaking hand with him. He is at Albert Einstein College of Medicine, and he is the one who created all the knockouts for linker histones and showed that in mammalian system, H1 is essential for survival, otherwise death happens. So with this, I will thank you all for listening, and I will take any question.
So we are highly thankful for your insightful and valuable presentation, and I strongly believe that this will definitely help our students as well as other participants to further use your valuable comments in their research, and they can do well in the field of research, and especially in this area which you have elaborated so well, sir. So we are highly thankful for your valuable contribution to this international conference, sir.
Thank you very much. Thank you for the invitation. Do I leave now?
Now I would like to invite Dr. Puneet Gandhi to please share your valuable presentation to our students as well as our participants in this conference. And ma'am, please enlighten us with your presentation.
Okay, a very warm good afternoon to everyone. Can I ask Dr. Rashmi to put on the presentation, please?
Ma'am, I'm sharing, just wait for the moment, ma'am. Sorry for that.
Sure. Okay. Can I go ahead?
Yes, ma'am. You can please proceed, ma'am. Okay. Ma'am, is it clearly visible to you also?
Yes, it is visible.
Ma'am, please, you may proceed, ma'am. Thank you so much.
Okay. So, COVID-19 has now been around for 6 months, and you must be wondering still, why do we need to talk about it? But I think by this time all of us know that this organism is around to stay. Even if the vaccine is there, being a virus, this organism is going to be within our systems for many more years to come. Can I have the next slide? Can I have the next slide, please?
Yes, ma'am.
So before we go into the biotechnology and the role of biotechnology in today's troubled times, I would just like to quickly take my audience through some myths and facts about COVID-19. Most of us are familiar with this, but I would just like to highlight a few things with this. For some time now, it is believed that the elderly and children are more at risk, but according to some studies in PubMed, there is a geographical variation in the age group that is being affected by COVID. The other myth was that when the infection started, it came to India, we were anticipating that with our summers round the corner, the infection will not be very severe. But according to the Journal of Hospital Infection, there are 10 studies internationally available now, which say that a minimum of 56 degrees Celsius is needed to inactivate this virus. All of us who work in the lab are aware of this fact that viruses need that temperature. So the sun exactly does not destroy it. Whatever is in the air does not remain at hot temperatures, but the suitable temperature would be at least a minimum of 60 degrees Celsius for 30 minutes, which can be obtained in a hot air oven in the lab or maybe just a commercial domestic oven. It was supposed to be airborne. We now know that the transmission is through cough and nasal droplets, and they stay in the air being in vaporized form for a few seconds. But this disease thankfully is not airborne. It is transmitted by human contact. Then there were rounds of news on the social media saying that vitamin C, D, zinc, and many such things could help cure infection. This again is not true. It helps only to enhance our immunity. Most of my being in a hospital, my colleagues and everybody has been taking a hot water bath, drinking hot water as prevention. They go home, they say it's a long routine. After working for such long hours these days, you have to go home, take a bath, change. Yes, changing your clothes is a good safety measure, but scientifically, since the virus activates at 56 degrees Celsius, the body temperature at all times remains at 37. It will only help to have any touch contact infection if we've had. Otherwise, hot water bath will not help, and drinking hot water will only help in case you have a throat infection. Use of sodium hypochlorite or alcohol on the body kills the virus. Both these things basically are used for surface disinfection. Sodium hypochlorite, a minimum of 0.5% is suitable. If you are dealing with biological samples, minimum 1% should be used. And as all of us from the medical or life sciences stream are aware, alcohol, a minimum of 70%, is a very good fixer of all kinds of microorganisms. So if you are using hand rubs and all, ensure that there is a minimum of 70 or 75% alcohol into your disinfectants. Then there was also a statement initially in March that consuming eggs, meat, fish, chicken also can cause virus infection, and a lot of birds were killed because of this. But this is not true. The infection is not through this host, and in fact, eggs being rich in protein for people who are not vegetarians, it is being given or being encouraged even by the medical fraternity. Can I have the next slide? Okay. The next slide, please. Yes, now actually the facts about COVID-19 infection. It infects our species regardless of race and ethnicity. The intensity of infection varies from person to person. Wearing a mask is most important, especially for frontline workers like us, or if you have a caregiver who's looking after an infected person. N95 mask is the suggested precaution to be used. Otherwise, for the general population, please use a triple layered mask when you are going to public places. Then this helps to a very great extent. It is also now known that COVID-19 in itself is not the only risk that we face. People with comorbidities like maybe diabetes, hypertension, and underlying kidney condition are at a greater risk. Why this is so, we will discuss when we move on to the structure of this virus. The other thing that we need to know is even if you are not into an urban location, even if you are in rural areas, please make sure that you have an RT-PCR test if the COVID-19 infection is to be detected, because without that, it is not a confirmation. Apart from that, the government and health agencies, the Ministry of Health has been giving out antigen tests which can confirm an asymptomatic status or an antibody test. There have been serological surveys in many states of the country. This basically, if it becomes positive, if it gives a positive indication for IgG, this test, then you have an immunity passport. You are lucky that you were infected and you did not come to know and you have passed on. But still, even in this condition, people who have been asymptomatic, I would like to take you along in the next few slides to know that we are still at a risk of some damage within our bodies and what should be taken care of. Can I have the next slide? Okay, so these are the general issuances from the WHO Health Organization, which says that even at low risk does not mean there is no risk. So we should be taking care of ourselves. The next slide, please. Yes. So far, all the studies say that as we were anticipating, children would be at a greater risk, but it seems their immunity is better than the ones who are in the 50 to 70 bracket. So children and adolescents till date have less of symptoms and have less of this infection. Next, please. According to WHO, as of date, most of the people who are down with this infection have also recovered or are recovering. So we don't have to fear this infection, but we need to know or make ourselves aware of how to deal with it. Next, please. It is very important when we are talking of an infectious organism which is creating a pandemic, that we are aware of the epidemiologic triad, as we call it in the health sector, in which not only the host, the causal agent, but also the environment is contributory. Can I have the next slide? Okay, so like I mentioned, the SARS-CoV-2, that is the novel coronavirus, and in this case we are the target, but the primary host is a bat, that all of us are aware. Apart from that, our surroundings, which are neat, clean and healthy, also would help us to avoid a lot of co-infections that come with this disease. Next, please. Can I have the next slide? These are the routes of infection. I think everybody is aware. There could be a direct contact or there could be an indirect contact. Next slide, please. In this, I would like to highlight that the interesting fact is initially a lot of expecting mothers were very worried when this COVID-19 infection started to spread across the country. But it has been clearly seen that during childbirth from the placenta there is no transmission, and there are very few registered cases in the country where the mother has infected the child. So the risk factor in this case is not through transmission, which we are aware. Can I have the next slide? Now coming to the virus itself. Why do we have to know this virus? The better we know our enemy, the better we will be able to handle it and to tackle it. In this slide, I would like to highlight two specific proteins: the spike protein of this virus, which is indicated with this green icon, and the membrane protein. These are the two things which normally bind to the receptors in our body and are creating a problem for us during its replication. Can I have the next slide? In the infectious disease process, there's the agent, there were the reservoir, which were bats. Now coming to the most important point for us, there is a portal of entry. For the human host, it is the nasal passage, and it is known to be excreted through urine and the fecal matter also. There have been a few studies which document that live virus has been found in human feces. The receptors for this into our body are ACE2, TMPRS2 and MPRO. These are the receptors which are present in our nasal passage, in lung, kidney and heart, and this is where the virus goes and binds. The mode of transmission we have already seen. The last important point in the disease process is the host immunity, and this is what creates the difference in the infection status, whether a person is asymptomatic or it is a moderate infection or it is severely asymptomatic. So apart from the age factor, the host immunity is what should be taken into consideration. Next, please. Actually, there are 5 phases of infection. Everybody must be aware of this. Incubation. Everybody knows that there is an incubation time from the entry of the pathogen into our body. Also, to be taken into consideration is a prodromal time. This is the interval between the actual invasion of the body and appearance of the first sign or symptom. Why I have highlighted this is this explains the difference of incubation times, or like we say, the quarantine times that we have seen over a period of last 6 months. There were people who were infected within a week of their contracting or coming in contact with a positive person. Symptoms have appeared after 14 days in certain cases, and sometimes as late as 21 or 28 days. So these are the phases of infection, and apart from the comorbidities that a person might have or might not have, the immunity of the said person plays a major role. May I have the next slide? So why do we say everybody knows about it? But I would like to say that as healthcare workers, we have found that it should not come from only the top. It is everybody's business, and we should all be looking into it. The upcoming week, October 14th to 20th, is celebrated as the International Infection Prevention Week. So all of us might take a pledge that we could contribute towards our immediate surroundings, at our places of work, at home. This kind of awareness is very important for us. Little points that everybody should know and contribute their bit about. Next slide, please. So when we come to talk of infection, there has to be an infection control policy. The guidelines have been many times given out by our health ministry, the Ayush ministry, individual institutes, and they keep on updating information as and when it comes in. But there are certain basic procedures of which we are all aware. Hand washing and keeping the workplace or your home and surrounding clean is the most important. This is what we call an infection control policy. Next slide, please. These are standard precautions that we take. Among all these that you would already be aware of, disinfection and waste disposal are two things that I would like to highlight. Like I mentioned earlier on at the start of the talk, that sodium hypochlorite or bleach as it is called in a layman's language is being used. So 0.5% bleach can be even made at home. It is commercially also available. It can be used to wipe surfaces to keep them clean, and hand washes. Wash with soap and water, that is equally good. And if you are traveling somewhere, then you can use an alcohol-based hand rub. Next, please. When we talk of a lot of infections, there is another aspect that we have been facing for the past few months, and that is people are also scared of coming to a hospital, because we know that nosocomial infections, or as we say hospital acquired infections, also can be there. You have come for a different kind of a consultation and there is a chance. But I would like to assure everybody that hospitals, healthcare facilities these days are taking a lot of precautions. Areas which are frequently visited by patients are regularly sanitized. Even our workplaces, there is a 3R circulatory protocol in place. That means every 3 hours, all the surfaces that are commonly touched are cleaned out, are sanitized, and then before we lock up for the day at night, again the areas are sanitized and closed. Next slide, please. Dealing with this infectious organism, we all know, and most of you must have gone through papers or in the social media, that the problem is that this virus can raise a cytokine storm. That means any microorganism, when it infects our body, raises an immune stimulus. But this virus can raise the cytokine levels, the pro-inflammatory cytokines like IL-6 in our body, TNF-alpha, to a very high level. And that is because this high cytokine level helps it to multiply even more. So this is one aspect of the virus that we need to keep in mind when we are developing prophylactics for it, when we are dealing with it for treatment, or when we are dealing with post-COVID infections. The other aspect is bacterial infections and fungal infections are equally important, although they are secondary. But the thing is that this virus brings down the immunity of our body, and it paves way for bacteria and fungi, which are opportunistic organisms, to come into our body. So we have seen that for many a times in a lot of cases where we have lost people is because not of the initial viral infection, but because of accompanying pneumonia in the lungs or certain other infections like sepsis is being caused, which is the cause of death. So this needs to be taken care of when we talk of later of developing vaccines or prophylactics. Next, please. Yes, it is a little scary for everyone to just walk into a hospital or a healthcare facility. But if you have any of these conditions, please do not delay. The reason being that these conditions accompanied by a silent COVID infection could cause problems. Next slide, please. The main culprit, that is our COVID-19 virus, is capable of causing acute respiratory infections. And accompanied with that are environmental conditions and a host of other factors like probably cigarette smoking, immune status. So an awareness regarding the contributory factors, or like the confounding factors as we say, is important when you are attending a person, maybe a family member who has a COVID infection. Next, please. For prevention and control, like we already have discussed, hand washing is the best way. Social distancing, if you can maintain, if you don't need to go out, please don't expose yourselves. But if you have to go out, take the necessary precautions, be alert, and then everybody these days is going about the normal business. We are out every day, going to work, getting things for the family. It is just even traveling for that matter. But I have seen that apart from authorities, people are, when they are careful, none of these risk factors would be involved. As far as food safety is concerned, if you are consuming food from a hygienic place, like I mentioned earlier, this virus is inactivated with heat. Then even food from an outsourced place should not be an issue. Just ensure that it is heated properly. In case this is especially true of sometimes of students who are out of their homes for studies and need to depend on areas, just ensure that your food is heated, well cooked, then it would be safe. Or maybe sometimes even we have to manage that way. Next, please. Okay, these are the standard safety measures that we take. The first one that I would like to highlight is Staphylococcus. These are organisms which could infect the human host along with the virus. And if you are suspected of having that infection, then the best thing is that go in for a proper disinfection of the place. The areas where the patient has been is normally cleaned and closed at least for 24 hours, a minimum of 1 hour and a maximum of 24 hours. Please also ensure that the housekeeping staff or the person or even your maids at home who are doing it are adequately protected, especially if they are doing it with their hands. Some kind of even plastic gloves, if they are not torn, are safe. They should be doing that. Or even when you're doing it yourself, use gloves and use clothes that can be discarded or kept away at least for 3 days, closed in a plastic bag. Be absolutely sure that whatever has been used by a patient is discarded properly. At home also, these days, if you use a specific, normally we use a yellow colored bag, which is indicative of a biohazard material. If not, then use a thick plastic bag and just inform your the collecting van which comes in for collecting the waste material, and they will discard it by incineration. That is the major thing that we need to keep it. Disinfection of our environment, our safety, and then the proper discard of waste material in case we have an infected family member or a colleague with us. Next slide, please. Yes, this is the challenge. It is very easy to say, but when you are actually into the system, we know that disinfection needs to be done very frequently. There are occupied spaces. You have to take your working times into consideration. Some people might like it. Some people just say, let it be. So, but whosoever is in charge, like we have in each of our departments, infection control teams. If you are at home, I'm sure there would be somebody to overlook these things, oversee these things. So apart from being a major challenge, I think this can be tackled if it is brought into a general routine. Next slide, please. When we have infections, even though we have policies in place, even though we have methods in place, sometimes we do end up with an infection breach. The control breach is there. Can I have the next slide? So there are just a few things that we need to keep in mind. The type of breach, whether it is leakage from biological material from some place, or a mask which is contaminated has not been disposed of properly. You need to review that if you are into an institute. Take a corrective action. Even when you are at home, you need to get your family members together just to tell them that this has happened, this is the precaution that we will be taking from now on. That is how infection breach needs to be tackled. And if you have an exposed patient with you, then you need to inform the concerned authorities so that proper treatment can be given. And maybe the rest of the people who were around, who were in probable contact, should be given the necessary prophylactic or prevention, something for prevention. Next slide, please. Now coming to the interesting part of something which is positive in this grim scenario. Biotechnology has suddenly come to the forefront. With all the things that are simultaneously going around, the major challenges that we face today is that we need the repurposing of a multi-target drug till the time we have a vaccine which can be approved for public use. I think all of you must be aware that about 110 candidate vaccines are being tried all over the globe. But till the time that a vaccine is declared safe, we need to work with the available medications. One of the initial ones was favipiravir, and these days remdesivir is being used. This is an antiviral drug, and it is being hoped that this antiviral drug, since already it is known to be safe for human use, it is being used to try and control the COVID-19 infection in persons who have the more severity of symptoms. Then comes our basic challenge, and that is the development of alternative prophylactics. At one time, hydroxychloroquine, or HCQ as we call it, has been given to healthcare workers and frontline workers. This is because this anti-malarial drug has been repurposed, since it can attach to the ACE2 receptor in our body and thereby prevent the virus from attaching to that receptor. But it is also known that HCQ unmonitored and sometimes in people can cause side effects. So other prophylactics are also being looked into. At this juncture, I would also like to mention that a lot of plant-based prophylactics molecules are being worked out by the Ministry of Ayush. And I think all of us at home are taking a lot of preventive teas with ginger, with tulsi, which is known to Giloy. A lot of things are being used. They are good. The only thing that you need to look into is that you don't have these antiviral Ayurvedic preparations on and on and on. The reason being that you boost your immunity, but there is something also called immunomodulation. The concept says that when you enhance your immunity, being non-infected, or if you are infected and the organism enhances your immune response, there comes a time when the infection is not there or when you are absolutely fine, that you need to bring this immunity stimulus to a plateau level, which is known as suppression. The reason being that a lot of pro-inflammatory cytokines, if this go beyond a threshold level in our body, that is also not good. So an immune modulation is important. You can take all these kadhas, but please get in touch with the proper Vaidya or with your medical practitioner so that a proper immune modulation also can be maintained. Next slide, please. As biotechnologists, I think all of us are very much into reading about the candidate vaccines. The first two I would like to mention, Covaxin and Zyco-D. These are indigenous to India. The first one has been produced by Bharat Biotech in collaboration with Indian Council of Medical Research and National Institute of Virology, Pune. Its trials, human trials, clinical trials are being conducted at 12 different hospitals. For the second one, the Zyco-D, it is less talked about, but it has been developed by Zydus Caldera, and it is believed to have a DNA and recombinant measles strain in the vaccine. Again, it is under phase 2 clinical trials. The third vaccine candidate is the one that has been made by Oxford University and AstraZeneca, and it has been permitted since it has been the Serum Institute of India at Pune is also a collaborating organization. Its trials are being held at 17 different sites in the country. And all of us, I think, are keeping our fingers crossed that these clinical trials, if they are successful, we are anticipating that a vaccine would be available somewhere by the start of the next year, that is in January 2021. Till that time, can I have the next slide? The need of the hour, where students and researchers into biotechnology have a big role to play. Even when we have a vaccine, this is going to continue for some time. We need a lot of you people, qualified, trained lab people for RT-PCR, for antibody testing, and a lot of our research fellows are helping us with different projects also, which are based on COVID research and for clinical trials. Even when we have the vaccine with us, there could be a chance that people may not be convinced to take the vaccine. Why I say so is because there are concerns that the vaccine, the immunity of the vaccine may not be forever. With one shot, we will not be able to negate the effect of the virus pandemic. As per studies available currently on the public domain, it has been observed that antibodies to this vaccine last for approximately 3 to 4 months. So whether this vaccine, whichever is declared to be useful for public use, whether it would have an efficacy of 3 months, 6 months or not, this only time will tell. So that so a vaccine also might not be the last resort. That is the reason all of us in the healthcare sector are still trying to make everybody aware of how to avoid an infection, and in case you have been infected, of how to deal with the symptoms, of how to ensure that the virus does not damage your body. The next concern could be that the virus may mutate. As we know, viruses keep mutating. So and we are a very big populous country. So by the time the virus mutates and by the time the vaccine comes up against a specific protein that would be targeted, it might also, there are chances it might be ineffective. Like I just discussed, immunity also from the vaccine could be an issue. And if and when everything goes in our favor, then the biggest challenge in our country is going to be distributing the vaccine among a population of 135 crores. The purpose of saying this was not to scare anyone, but to be alert, to know your body, to know how the virus works, to know how our body responds, and to take good care of ourselves. In general, good hygiene practices, healthy eating practices have been taken up by everyone. So there's no single solution to controlling the spread of this virus. An effective infection control policy, whether it is home, workplace, anywhere that you are, is most important. Government agencies, healthcare agencies, and workers, communities are into it. But the main thing that we need to deal with is that we need to address the risk factors that are associated with it, which I've dealt with. Immunity at the prophylactic level is very important. If you're taking something, just because somebody says that taking a potion, a tea, a kadha, day in and day out will help, that is not the actual fact. If you have been taking it for some time, consult a medical practitioner. See that your body immunity levels are okay. That can be tested absolutely in a lab by giving your blood sample, and that would be the best way to deal with this infection, this virus, this enemy for the times to come. Next slide, please. I think that brings me to the end of my presentation. Thank you so much for a patient listening. I hope I have been able to convince most of you that this infection can be taken in stride. It's just that we need to address our behaviors. Thank you.
We are extremely thankful to Puneet Gandhi ma'am for covering such a significant topic as we all are facing with this pandemic. So I strongly believe that this presentation will definitely clarify all the myths associated with this disease, and also this will motivate our young biotechnologists to further extend their contribution to the society, either through their research or either making the people aware about this disease, as Puneet Gandhi ma'am has explained each and every minute detail about this disease very significantly. So I'm heartily thankful to you, ma'am, for sharing such an insightful presentation to all of us. Thank you so much, ma'am.
Thank you. Thank you.
Now, I would like to invite Mr. Ankit Kumar, sir, who is the Deputy Head of the Department of Biotechnology, to further extend vote of thanks to all the eminent speakers Ankit sir, please.
Okay, thank you, Dr. Pratibha. I take this opportunity to heartily thankful to Dr. N.G. Lakshmi Narayan sir, Dr. Lakshmi Narayan Mishra, and Dr. Puneet Gandhi ma'am for their valuable time and very interesting and informative talks. Although due to technical glitches, we could not completely hear Dr. N.G. Lakshmi Narayan sir and thus could not extract all the knowledge that he had to enlighten us with. The talk had to be cut short. Nevertheless, we thank you, sir, for trying so hard to connect with us, and we would certainly like to invite you at NIET when situation permits. And also thanks to Lakshmi Narayan sir and Puneet Gandhi ma'am for the wonderful session. And this session was in alignment with the mission of our department, that is to impart the youth with the knowledge of analytical approach, research aptitude, and technology development in the field of life science and biotechnology. I am certain that these sessions will be beneficial for all the participants. And I again would like to thank all the speakers and participants. Thank you so much.
Again, I am thankful to all of you. Now the session is over and hope we'll meet soon.